Our second speaker of the day is Dr. Robert Schwartz. Dr. Schwartz was educated in Vienna and stayed in Vienna until the time of his postdoc, at which time he moved to Baltimore to do his postdoc with Joe Coyle. And then he did his PhD he did his postdoc with Joe Coyle. And then he went to Sweden and worked with Kjell Fuchse and Thomas Hochfeld for a second. Anyway, I may have this wrong. Right after that, you would think Robbie has a fairly simple, because he came to Baltimore, and then he came back to join the faculty at the University of Maryland at the Maryland Psychiatric Research Center and has stayed there for his whole career. Robbie has studied the kynurenic acid system and has, with a focus on that system, has learned more about the system than anybody ever, but in addition to that has spawned a number of areas of research that he'll tell us about today. Dr. Schwartz, dysfunction of kynurenic acid in major psychiatric diseases.
I'll do it. Okay, thank you very, very much, Carol. You got it almost right. My talk is going to be different conceptually from the really beautiful talk that Yasmin just gave us. I'm a biochemist by training, and just briefly about the history. So I wanted to learn about neuroscience, so that's why I got into working together with psychiatrists, and I've done this all my life, but I'm still a biochemist at heart. So the story I'm going to tell you is really starting from bottom up, if you wish. And I'm going to tell you about a compound called kynurenic acid, which you just heard about, and I'm going to highlight just a few items for time reasons. There's a number of people who are working on this now, and I apologize to people whose work I do not mention today, but I'm not neglecting it. It's just for time reasons, and just to highlight a few issues that are hopefully of interest to this audience. So the story started in 1853. This is not a mistake. This is the whole paper that was published in 1853 by Justus von Liebig. It's in German, and it says über Kynurensäure, which means about kynurenic acid. That's the only paper he ever published on kynurenic acid. He was a polymath, very, very famous person, and many of you may have heard of him. But he discovered that this compound, which he called kynurenic acid for this reason, is present in high concentrations in dog urine. It crystallizes, it's such high concentration. And then many years passed, and people ignored this for a long time. And what we know today, though, is that kynurenic acid here is part of the tryptophan catabolic pathway. And just to remind us, as people who are in biological psychiatry, for many years, for good reason, we always focused on serotonin, maybe on melatonin. Only 5% of the essential amino acid tryptophan that we ingest gets metabolized to serotonin. 95%, in contrast, get metabolized along this so-called kynurenine pathway. This has been well established mainly in the first half of the 20th century, outside the brain. There was always this in most cases, unspoken implication that this pathway does not exist in the brain. It's all about serotonin in the brain. Turns out that it is very, very interesting in the brain, and there are several compounds which I'm not going to talk about today, including kynuric acid, but the others, xanthorenic acid, stearodoxanthorenic, and so on, particularly quinolinic acid you may have heard of, which are neuroactive compounds which are part of this pathway and you know very, very little about. So if any of you are interested in finding a new compound you want to work on that hardly anybody works on yet, there's plenty of them here. But today I'm going to talk about kynuric acid, and the reason for that is that in the early 1980s, this is the time when, believe it or not, there are people still out there discussing if glutamate is a neurotransmitter. They just found those ionotropic, as we call them now, ionotropic glutamate receptors, which were called Kynurenic acid, AMPA receptor, quisqualate receptor, and NMDA receptors, which are all classic receptors now. But the interesting thing is that an electrophysiologist in London showed that kynurenic acid blocks all those receptors at high concentrations, though. And then people started to get interested in it. They looked at kynuric acid as a tool to block those ionotropic receptors, and it's been widely used, and it's still widely used just as a tool. Then it was shown that by Edson Albuquerque at the University of Maryland that kynurenic acid is also an antagonist, a non-competitive antagonist of the alpha-7 nicotinic receptor. And it also, at concentrations which are lower than those which block the ionotropic receptor, it also blocks, maybe specifically or preferentially, I should say, the glycine coagonist site of the NMDA receptor. And then we found that kynuric acid is actually present in the brain, which was not even known at that point. And kynuric acid is present in the human brain in higher concentrations than in the rodent brain, but it's present in all the mammalian brains we've looked so far, and other people have looked, including monkeys, of course. So it's about that range, and this slide is something to remember when we talk about potential implications or roles of kynurenic acid in brain function and dysfunction. So at the endogenous concentrations, and remember, these are brain tissue concentrations we're talking about. So we all know the problem with extrapolating brain tissue concentrations to synaptic concentrations or extracellular concentrations. So just keep in mind that kynuric acid it can act as an inhibitor of glutamate receptors, alpha-7 receptors, and it turns out there are more receptors that kynuric acid targets. So this is still a work in progress. So again, as a biochemist interested in enzymes in particular, we looked at that, we found out that there's one enzyme called kynurenine aminotransferase 2, or KAT2, which I'm going to talk about for the rest of my presentation, which is primarily responsible for the production, for the de novo production of kynuric acid in the mammalian brain. So we purified the enzyme to homogeneity, made antibodies, classic way, and found that kynuric acid synthesis takes place in astrocytes. So now we're dealing with a compound that is present in the brain, acts at receptors, which we are all more or less interested in, and it's not a neurotransmitter, it's a glial transmitter or something like that. So the concept is very, very simple then, and we like simple concepts. We're getting to Yasmin's level probably in about 30 years. So kynurenine, which is the precursor of kynuric acid, gets into the brain easily through a transporter, and then it gets taken up in astrocytes. Details are all published. And then this enzyme KT2 produces very rapidly kynuric acid, which then can get out of the astrocyte and for potential interactions with, as I said before, alpha-7 and NMDA receptors. So just keep that in mind, because that's really as far as we got, we as a field at this point. But it's always worth remembering when you look at an EM picture, how closely the astrocytes are engulfing the synaptic terminals and the synapse in general. So I put this KYNA, kynuric acid, in there just to remind ourselves that when kynuric acid is produced, and it is produced very quickly, as I just said, it has very, very rapid access to any receptors, including alpha-7 receptors and NMDA receptors, maybe others, on neurons. So again, conceptually, one of the things we tested, and I'll show you just one data slide on that, is that, so this is the astrocyte here, and this is a glutamatergic nerve terminal, and we know from many people's work, maybe some of you, that throughout the brain, alpha-7 receptors are very, very frequently located presynaptically on glutamatergic terminals and regulate glutamate release. So if kynurenic acid is an endogenous antagonist of the alpha-7 receptor, then you would assume that it affects glutamate release. And you can do these studies using, for example, microdialysis in unanesthetized animals, which we did. And so this is extracellular glutamate measured here, %. So what you see when you locally infuse for a couple hours, at low concentrations, this is essentially endogenous concentrations, maybe in the range. 100 nanomolar of kynurenic acid through the microdialysis probe, we call this reverse dialysis, as many of you know, then the extracellular glutamate levels go down. And this can be blocked by an alpha-7 agonist. And again, we don't have to talk too much about galantamine. I know that it's not only an alpha-7 antagonist, but it is certainly the concentration we used here, and we showed this with other alpha-7 agonists, the same result. So the effect of kynuric acid on glutamate, so it reduces extra glutamate, is mediated by alpha-7 receptors, probably those that are sitting presynaptically on the nerve terminal. And it turns out, when you do more studies, that kynuric acid not only affects extracellular glutamate, it affects essentially, well, not essentially, it affects several of the neurotransmitters that we've been working on for many, many years, all of us. And so I like this cartoon, which was made by my friend in Sweden, Sophie Erhardt, and her colleagues, actually. And so it turns out that we start to think about kynuric acid in astrocytes regulating in different parts of the brain. This was done not only in the prefrontal cortex, which I just showed you, also in the striatum, hippocampus, cerebellum. So wherever we looked so far, we see basically the same thing with some variation. That it affects those neurotransmitters. And in all cases, these cases, when you increase kynuric acid levels just a bit, and this happens when you ingest tryptophan, for example, we'll come back to that, then those neurotransmitters are affected. Now, another study which is of interesting, I think the people are interested in biological psychiatry, it was done by Kuei Zhang, who is an electrophysiologist in Chicago. And what he does, he stimulates, this is in anesthetized animals, he stimulates the ventral hippocampus, and we're all interested in the connection between the ventral hippocampus and the prefrontal cortex for reasons which I don't have to explain here. And he found that when you then infuse kynurenic acid, again at more or less endogenous concentrations, he did a concentration curve, which I'm not showing you here, then it turns out that the local field potential in the prefrontal cortex is affected by kynurenic acid, but it's done in a frequency-dependent manner. So only the higher frequency kynuric acid does this. So kynuric acid in the prefrontal cortex does something that is, again, of interest to people who are interested in biological psychiatry. And we're also very interested in behavior. So without going into any details, it was done by many people. The bottom line here is that when you increase, this is done in mice or rats. I showed just a summary of rats. There's dozens of papers out now, always more or less saying the same thing, that when you increase kynurenic acid levels just a bit in the brain, you get cognitive dysfunctions. And mainly using, of course, all the outcome measures, behavioral outcome measures that we're all using with our limitations, that are maybe informative to what's going on in people with schizophrenia. So we start to think about potential implications for schizophrenia, and I should also mention that the place I'm working at, the Maryland Psychiatric Research Center, is heavily focused on schizophrenia. So we're a translational place, very much, doing that, and have been done that for many, many years. So, how about kynuric acid and schizophrenia? Now, I don't show the slide that Yasmin showed, but we also have a brain collection at the Maryland Psychiatric Research Center, which actually our president and myself started many, many years ago. So what we can do is then we can just measure kynuric acid levels in people with schizophrenia. And this was done over and over again. I'll just show you one slide, which is sort of leading to the point I want to make. And it shows that in Brodmann areas 9 and 10, kynurenic acid levels are increased. Not a lot, but they're increased. And this has been again shown in other brain regions as well, cortical regions as well as the neostratum. What is particularly interesting, what I want to point out here, is that the enzyme KMO, and I'm coming back to that in a moment to explain this, in the same tissues, this exact same tissue samples, this enzyme is decreased. Now, why is this potentially interesting? Because kynurenine 3-monooxygenase, or KMO, which I just showed you is decreased in schizophrenia, is the enzyme that is placed in the pivotal part of the kynurenine pathway. So what happens, and again, without showing too many hard data here, is that when you reduce in animals KMO, you can do this pharmacologically, you can do this genetically, the substrate kynurenine is increased and then gets taken up, as I showed you before, into astrocytes, and kynuric acid gets up. So we have a mechanism here, a potential mechanism that explains why kynuric acid levels may be increased in the brain of people with schizophrenia. So one of the things that you can do, of course, is that you can make animals, mice in this case, which have a KMO knockout. And it turns out, and this was published just a few months ago, turns out that they have abnormalities in various behavioral outcome measures, which are again relevant for the study of schizophrenia. And also, as we just, this is just very new data, not published yet, with Jörn Engberg at the Karolinska Institute, we found that there's also an increased activity of VTA dopamine neurons in those mice. So a lot of indications that these mice, which also have morphological abnormalities, which are reminiscent or what's happening in schizophrenia, this is something that has to be looked at very, very carefully, and we and others are in the process of doing that. So the link between this enzyme also indicates that maybe we should look into the genetics of this enzyme, and without giving you details here again for time reasons, is that there are not only SNPs, but there's a developing story on the genetics of KMO. And as far as we know, there's only one KMO gene. There's no KMO 1 and 2 and 3, which makes it a bit easier. I think we have to stay tuned and see what comes out of this. But I find it very interesting, and sorry, it's not very attractive to say this in a talk that, you know, I have interesting data and don't show you any. But it's just a bit premature. But we should all be aware that this enzyme KMO is something that's of potential interest for maybe subgrouping people with schizophrenia and maybe more. So let me just spend a couple minutes on one issue which is of great interest to all of us. And we all know that schizophrenia is a developmental disorder. So one of the things we can do with the kynuric acid story in mind is we can see what happens when you increase kynuric acid prenatally in the fetal brain. And this can be done very, very nicely. This was pioneered by Anna Poczewawczyk in my laboratory several years ago. So you can feed the precursor of kynuric acid, kynurenine, to rats. We have always done this with mice with essentially the same result. What happens is, what happens very quickly is that kynurenine, this is the compound that you feed, goes up, of course, in the blood, but also kynuric acid levels go up, and this is what we really wanted. So at the time when the animals are born, they have higher kynuric acid levels in their brain. Now, then afterwards, we wait for a couple of months, and they are just treated with normal chow, fed normal chow. And interestingly, when we look after 2 months, we've done this over, we didn't believe it in the beginning. We said, why? The kynuric acid levels, including this is the tissue levels, extracellular levels included, in the cortex and in the hippocampus also, of these animals, which we call equine animals, embryonic kynurenine-treated animals, are increased. So this looks pretty much what you see in schizophrenia. I'm not saying there's necessarily a cause-effect, but something is going on in those 2 months that eventually leads to an increase in kynuric acid. What I didn't show you is that when you look earlier, you don't see this increase. So something is happening here. And one of the things, of course, we wanted to see if those prenatally treated animals are maybe behaviorally abnormal, and they are. And just show you one paradigm, the passive avoidance paradigm, which I'm sure all of you are familiar with. Animals like to go into the dark side of the box, and they get a shock there, and they remember that the next day. And normally, in the first day, they go in within 10 to 20 seconds. The second day, just the day after, it takes them 2.5 minutes to go in there because they remember this was a bad place to be, to get a shock there. Those equine animals do not remember. And I'll explain this in a moment, why I'm saying remember. So they are abnormal in this paradigm. They're also abnormal in other cognitive paradigms. So one of the things I just mentioned, the only thing I'm going to mention in terms of genetics, because we're going in that direction quite a bit, and I told you about looking at the human brain. But of course, you can also do things in the mouse. As I just showed you, we made KMO knockout mice. We also made KMO heterozygous KMO mice. And what I show here, it's a bit complicated, but please bear with me. So the dotted lines here are basal levels of kynurenic acid and also other metabolites of the pathway. Kynurenine, kynurenic acid, quinolinic, anyway, and others. So the baseline, this is prenatally, baseline doesn't change in the heterozygous KMO knockout mice. But when you treat them with kynurenine prenatally, when you stress them, we did that also, then you see a difference that the ones with less KMO activity react more to a prenatal stimulus. And you see increases in kynuric acid and kynurenine and reductions in the other part of the pathway. So it seems that the pathway shifts somehow in those heterozygous animals, and it only comes out when you stimulate. So it's very, if you think translationally, I don't have to explain this, I'm sure. So this could be very, very relevant. So we're looking very, very carefully in those heterozygous KMO knockout mice. Now, the last part of my talk, I have a few more minutes, right? I want to talk about the reason I guess I was invited to give this talk, and that is, are there implications for treatment? And I'm trying to be rather quick. It's very, very simple, the idea. Let's block KT2 and see what happens. And years ago, we made KT2 knockout mice together with Dan Tagley at NINDS at that time. And just to summarize, without going into detail, we found that when you reduce endogenous kynuric acid, this is what happens when you have a KT2 knockout, you increase extracellular glutamate, and you increase hippocampal plasticity, and you increase cognitive behavior. So that's what we were looking for, and we were very, very careful before we published this, and because it had a lot of implications. So less kynuric acid may be a good thing when you want to improve cognition. Where we stand now is that we want to get orally active KT2 inhibitors, of course. This is just a work in progress. We have some that work very nicely in animals. This is one of them, and this just shows that it's active. When you give kynurenine, kynuric acid goes up. When you give a KT2 inhibitor, it doesn't happen anymore. Interestingly, also pharmacologically in this case, as I showed you with the KT2 knockouts before, you reduce kynuric acid and you increase extracellular glutamate. So the exact opposite of what I showed you before. So again, show you this slide. In this case, though, it is not when you increase kynuric acid that the neurotransmitter levels go down. Now we found, this was done with several people, I'll show you the names in a moment, when you reduce kynuric acid levels in the astrocytes, so you block its synthesis, then you increase the extracellular levels of those neurotransmitters, all 4 of them. So again, interesting conceptually, and we think, of course, interesting for people who are interested in biological psychiatry. Just one slide to show you the effect on cognition, and that this is maybe a bit complicated, but not so much. I showed you before, it's against the passive avoidance paradigm, showing that if we focus on this, these are the control animals on the second day, so they remember not to go into the dark box because they had a shock there the day before. And the prenatally Kynurenic treated animals, they don't remember. I showed you this before. Now, if you give a KT2 inhibitor on day 1 to these animals, they remember very well. So that's why I said remember before. When you give the KT2 inhibitor on the second day, it doesn't have any effect. It looks just like this. So we're starting to learn about the role of endogenous kynurenic acid in various cognitive functions. Just one example. And finally, we are starting to translate this into humans. And what is known, this is an ex-postdoc of mine in Japan who showed that when you give tryptophan to animals, then, as I told you before, kynurenine can get into the brain, goes in astrocytes. The important part here is that when you give tryptophan, then the brain concentrations of kynurenine and kynurenic acid increase. And this is what I want you to remember, because you cannot do this in humans. But in animals, it's very, very clearly shown. So we started to give it to humans. So we give tryptophan to humans, which had been done for decades. So it's safe, and it wasn't a problem with approval. And what we've shown, and this was done by Laura Rowland at the MPRC, as you can see, is that visual learning and verbal learning is impaired, and we are following this up now. And of course, this is a paradigm that we're using to maybe apply KT2 inhibitors and see what happens. So in summary, I don't know, do I have 3 more minutes? Yes. Okay. So just to summarize, because I have one more thing I want to show you. I wasn't sure if I have enough time. The kynuric acid story, I probably don't have to repeat too much here. Fluctuations in endogenous kynuric acid in the brain have effect on neurotransmitters, up and down. So we think it controls, that's why the cartoon was so cute, controls extracellular glutamate, GABA, and also dopamine. We did quite a bit on that. And even relatively minor elevations, either prenatally or early in development or later in life, impairs cognition. And again, we can talk about this quite a bit. What does cognition mean and so forth? It is a target for intervention. So let me just do a couple of minutes worth on this. I said I think the title was not about schizophrenia, it was about psychiatric diseases. So I want to just give you an example of how the work we've been doing, and of course many others have been doing, maybe also applied to major depressive disorders. And this was done, this started a long, long time ago. It's one of those things where you get an idea and then you don't follow up. And so, but what was found in the late 1980s is that this compound that probably several of you know about, 7-chlorokynurenic acid, is a very selective inhibitor of the glycine coagonist site of the NMDA receptor. So we thought, this is an exogenous compound, man-made compound. So what if we make a precursor of this compound, which may use the endogenous mechanism that makes kynurenic acid out of kynurenine. So we made this compound, 4-chlorokynurenine, and indeed, it makes very nicely 7-chlorokynuric acid. And as you see here, just schematically, exactly the same way, this could have been kynurenine, kynurenic acid, glycine receptor. Right? But it doesn't act at alpha-7 receptors. And of course, we all know about the current controversies about how does ketamine work as a rapid-acting antidepressant. I'm not going to get into this. Other people have done this and will do this again in this group, in this meeting, and everywhere else, it seems. But one of the possibilities is still that NMDA receptors are seriously involved in the rapid antidepressant effects of ketamine. So what we wanted to see, and this was done together with Todd Gould and Panos Zanos at the University of Maryland, we wanted to see if 4-chlorokynurenine administration is effective in antidepressant models. And this is done in mice. And I'll show you just one slide for time reasons again. And we published this a couple of years ago. So it works beautifully. It works just like ketamine. So short-term effects, and 7 days later you see it also. So this is something now that is, again, I'm just trying to mimic and keep up with Yasmin. So this is now getting to a stage that is really translational. And Carlos Zárate, who all of you know or know of, is doing a phase 2 study right now at NIMH, and we're all waiting for the results. We don't know, but supposedly soon, but I guess that's what you always hear. So it's a double-blind crossover study, and we'll see if 4-chlorokynurinin is an antidepressant in humans. So stay tuned, please, and I'll tell you more maybe next time. So thank you very much for your attention. I just, well, I don't have all the nice pictures that Yasmin had, but you know, one doesn't do this alone, and this is just, I use the word current because I've been around for a while, and there's many more people that were involved in this whole story, and there's a number of my clinical colleagues, mainly at the MPRC, who are very much involved in this now and try to have a translational aspect that to the whole story, which again, hopefully I'll be able to tell you more before too long. So thank you very much.
Thank you, Robbie. There's time for one question.
That was fascinating. Thank you. Would you please comment on the impact of your work on cognitive changes in normal aging and what impact kynuric acid research might have?
So is the question on normal
aging deficits?
In normal
humans.
In normal humans, we don't know yet, because as I had on one of the slides, you know, we hope that where's the hope? We hope that a KT2 inhibitor will be available for human trials soon. We had really anticipated we'd be done this one without going into details, which I'm not allowed to do. But there's one company that is really has done the talks, and before we give a KT2 inhibitor to humans, we just don't know. The one thing, I do have a couple of slides, and this is a very contentious issue. I just mentioned this. I almost hesitate knowing who's knowledge to talk about N-acetylcysteine. So there's very mixed feelings about N-acetylcysteine and its mechanism of action. I'm very much aware of this. Turns out that, but there are indications, I think, that many of you will probably agree with, that N-acetylcysteine has pro-cognitive effects. Also in people with schizophrenia. So it turns out, well, let me show you. I have to do this. One slide. Oh, it's gone. So N-acetylcysteine is a very potent KT2 inhibitor. So we are starting to think about N-acetylcysteine as a tool, at least, although it does many other things we are aware of, as a tool before we have a KT2 inhibitor to see if the effect of N-acetylcysteine, also in normals, of course, is somehow related to reducing kynuric acid levels. It's not an easy test to do, as you can imagine. So just want to say it. Sorry, long answer.
So join me in thanking Dr. Schwartz for his talk.